So let's solve question number 186 until 190. Your question number 180 is which one is correct about bivalent chromosome? So you know bivalent chromosomes are chromosomes which are formed because of pairing of the homologous chromosome, right? That is the paternal chromosome and the maternal chromosome during the crossing over, right? The crossing over happens during the prophase one. Okay, so let's mark them. What are those structures? So it's a centromere, centromere. These are sister chromatids. These and these are sister chromatid, and this one and this one are homologous chromosomes. Okay, so based on that, let's compare the options. So first option is bivalence are tetrad. Yeah, because there are 4 chromatids. Okay, this is true. A bivalent means 4 chromatids and 2 centromere. This is also true. You can see centromere 1 and 2 here. Then one bivalent consists of 2 homologous chromosomes. This is also true. Okay, bivalent formation occurs zygotine. This is also true. Leptotene and zygotine are short-lived. This is also true. So here all of these are true. So your option will be answer A. The next one is, which one of the following properties of cardiac muscles are correct? These are muscles of the heart. Yes, these are muscles of the heart. It is true. These are non-striated. It is false. They are striated. These are involuntary in their action. This is also true. These are, now this statement is wrong. Okay. These are controlled by the nervous system directly, not directly. So this is wrong. Cardiac muscles are unbranched. It is wrong. They are branched. So the correct one is the statement number 1 and statement number 3. So your will be option A. Answer A. Then question number 188. Okay, epinephrine. Okay, no, epinephrine is released by the adrenal medulla. Okay, so statement 1 itself says that whether it's released by adrenal medulla, this is true. This is true. Okay, released by parasympathetic fiber. That's wrong. They are released by the sympathetic fiber. Increases the heart rate. It is true. They increase the heart rate. Decrease blood pressure. It's false. They increase the blood pressure. So statement number 4 and 2 are incorrect, whereas 1 and 4 are correct. Sorry, statement number 1 and 3 are correct. Okay, 1 is correct and 3 is correct. So your option will be option B. Answer B. So this is question 189. Okay, statement related to human insulin are given below. Which statements are correct about genetically engineered insulin? Okay, so statements are given. You need to find which is correct for with respect to genetically engineered insulin, or this is known as humulin. Okay, humulin. Okay, the pro-hormone insulin has extra stretch of C-peptide. This is true for human insulin, but not true for humulin. A peptide and B peptide chains of insulins were produced separately in E. coli, extracted and combined by creating disulfide bond between them. This is true. Insulin used for treating diabetes was extracted from cattle and pigs. This is not correct because we are talking about humulin. Okay. Pro-hormone insulin needs to be processed for converting into matured and functional hormone. This is true for insulin, not for humulin. Okay. Some patients develop allergic reaction to the foreign insulin. Okay. No, this is not true. Okay. With respect to the humulin. Okay. So choose. So here only option statement number 2 is correct. Okay. So we'll go for option A. So here answer will be A. Now you may make a confusion thinking of the insulin and humulin. We are not talking about insulin. We are talking about humulin. Okay. With respect to that, we can revisit the options. Okay. Then question number 190. Okay. Question number 190 asks you the sequence of method, okay, used for creating or doing the inserting the DNA inside an E. coli. Okay. So let's check. Okay. So the sequence is, first we need to treat them with the divalent cation. Basically, we use calcium. And reason is that, okay, you know the plasma membrane, right? Plasma membrane contain this is phospholipid. Okay. So this they are negative. Okay. Isn't it? They are negative. Okay. DNA, okay, which we are want to insert inside the cell. This is also negative. So if it come near the membrane, okay, there will be a repulsion. So we want to neutralize it. Okay. So we treat the bacterial cell, okay, with divalent cation. Basically, here we will use calcium ion. We use calcium chloride. When you add them into solution, it dissociate into calcium ion, okay, and chloride ion. Okay, they dissociate, right? Okay. Then calcium goes and binds here on the cell surface. Calcium binds. So calcium create positivity. DNA is negative. DNA come near the cell, bacterial cell. Okay. Then we what we do? Then we incubate them in ice. Okay, in ice, cold water, ice. We incubate them, and then okay, we incubate both the DNA. Okay, that is the foreign DNA as a cell. Then we put them into a heat shock. What heat shock does? Heat shock open the pores and increase the efficiency to uptake the DNA. And because of the pore, the DNA gets enter into the cell. Then we keep back them into ice to close the pore. Okay, so the DNA get locked inside the cell. So that is the method. Okay, so let's see which treatment okay align with what we just spoke of. Treatment with divalent cation. Okay, followed by incubation of cells with recombinant DNA on ice. Okay, heat shock. Then placing. So this treatment is correct. Okay, so your option will be or answer will be option A. So this much for this particular session. Okay, or this particular video. We'll meet again in next video.